The effect of flavivirus immunity on Zika. Zika is a flavivirus transmitted primarily through the bite of an infected mosquito. Zika is endemic to specific parts of Africa and Asia. It remained relatively obscure after discovery in 1947, up until a massive outbreak in 2015. It quickly spread to other countries in South and Central America and is now present in the United States. The virus persists in human tissues for long periods of time, leading to sexually mediated transmission, potential infertility, and pregnancy complications. Dengue virus is the leading cause of illness and death in the tropics and subtropics of the world, and is endemic in similar regions as Zika. Both are RNA viruses, which usually lack proofreading, leading to high mutation rates, making them capable of rapidly outmaneuvering the host immune system. Researchers are exploring the possibility of antibodies against other flavivirus-enhancing Zika pathogenesis. Antibody-dependent enhancement of different dengue viruses has been shown to correlate with increased severity of Zika. While most symptoms of Zika are mild fever, rash, and pain at worst, a dangerous potential outcome is the development of microcephaly in fetuses. Microcephaly is a rare condition that causes 7 per 10,000 babies to be born with undersized heads and underdeveloped brains. About 1 in 20 babies in U.S. territories have Zika-associated birth defects. The consequences of microcephaly include vision and hearing problems and other developmental issues. The risk of microcephaly is a worldwide threat because it is a lifelong condition with no known cure or standard treatment. Current treatments of Zika and dengue also focus on symptom management. However, there are several ongoing clinical trials for Zika vaccines, although their infection mechanisms are not completely understood. Flaviviruses such as Zika and dengue are positively charged RNA viruses. Their structure consists of a genome enclosed by multiple copies of a capsid protein, further enclosed in a lipid membrane which anchors membrane and envelope proteins. During the life cycle of a flavivirus, there are 3 major stages. The first stage is immaturity, where the virus is not infectious. During this stage, the virus is assembled in the endoplasmic reticulum of a newly infected cell before it is transported through the secretory pathway. The second stage of the life cycle is virus maturity, where the particle moves into the trans-Golgi network. Here, the virus goes through conformational changes, allowing exposure of the cleavage site for precursors on membrane proteins. These are then cleaved by the protease furin. The virus is next released into the extracellular fluid, where the precursors completely dissociate, exposing fusion loops and priming the virus for endosomal fusion with another host cell. It is thought that electrostatic interactions between the positively charged fusion loops and negatively charged glycosaminoglycans, or GAGs, on the host cells allow the virus particles to concentrate at the surface of the target cell before entering. It is also believed that the amino acids that surround the glycosylation site in each of the virus's envelope proteins is what primarily differentiates flaviviruses. This site is unique in Zika and may function as an attachment site to host cells, as well as a modulator for tropism. When a host is infected with a virus, the immune system mounts humoral and cell-mediated responses. As part of the humoral response, B cells secrete antibodies that circulate throughout the body. We will be talking about the antibody type immunoglobulin G, or IgG. IgG is a Y-shaped molecule made of 4 polypeptide subunits. The N-terminus is the region specific to antigens. At their C-terminus, antibodies can be recognized by Fc receptors of specific cell types. Antibody-dependent enhancement, or ADE, occurs when a previously infected host undergoes a secondary infection with a similar virus. As dengue and Zika are both similar flaviviruses, ADE is a complication that arises when an individual is infected first with dengue, then Zika. The immune response to the primary infection leaves behind memory B cells that are specific for dengue. Secondary infection causes these B cells to recognize and proliferate plasma cells, producing an abundance of antibodies specific to the first virus. This exacerbates the infection because these antibodies are able to bind to the virus but can't neutralize the threat. Fc receptors on monocytes recognize the Fc portion of the antibodies and then phagocytose the antibody-virus complex. This infects the cell and leads to an increase in viral load. ADE can also cause cytotoxic T cells to be overstimulated and secrete an overabundance of cytokines. In high volume, they can cause tissue damage and serious inflammation, therefore compounding the effects of the virus. Ultimately, ADE results in increased viral load, viremia, and increased morbidity. For future research on Zika virus, the National Institute of Allergy and Infectious Diseases is working with biotechnology industries and sponsoring researchers on developing sensitive, specific, and rapid clinical diagnostic tests for Zika virus. In addition, researchers are creating treatments for Zika virus and broad-spectrum antiviral drugs that will be effective against multiple flaviviruses. They're also developing and testing vaccines to prevent Zika virus infection. Finally, these vaccines have to be carefully manufactured to avoid the effects of ADE.